Hi everybody. Have you ever wondered if there is a way how to read our DNA and then use it to our benefit? If yes, then I wrote a short article on my blog sharing with people, and it's called How to Read Our Life Code. So let's kick off. Let's start with a little of history. A very interesting fact is that it took 32 years to fully sequence human genome, also called DNA. It all started with so-called Human Genome Project in 1990s, which completed in 2003. But by then we sequenced just 92% of human DNA, and then sequencing of the remaining 8% took us another 19 years, so in total 32 years. And thankfully it completed in 2022. Thanks to this achievement, today we can sequence DNA in parallel and under one day, which is amazing if we compare it against 32 years. So what is this substance called DNA? It's deoxyribonucleic acid. In other words, it's a molecule that carries the genetic information for development and functioning of an organism. So you can imagine it like some instructions how to, for example, create some amino acid or how to transcribe RNA. So we sequence the full human DNA and so what? Now, thanks to the very powerful computers, AI, and machine learning, and also parallel processing during the sequencing, we can try to find patterns inside our DNA connected with health records. So if we collect enough samples, and we know that, for example, this amount of people has high blood pressure, and then we let the AI and machine learning crunch through the data, we can figure out that if we have these genes inside our DNA, there is a higher probability of evolving the high blood pressure. And then we can take proactive actions to prevent it. There are two scientific areas which study DNA, or how the DNA transfers the traits in between people, or how it influences the cells. One is called genetics, which is studying the genes by itself. And then to make things more interesting, there is also an area called epigenetics, which studies something like on top of genetics. So it's not really about changing the genes, but it's rather about changing the expression of genes. So you may figure out that you have, for example, gene or series of genes that may increase your probability of evolving high blood pressure, but because you exercise and you eat well, thanks to epigenetics, the expression of these genes may be suppressed. So can we use today this knowledge to benefit us? Yes, we can. It all begins with so-called extraction and sequencing of DNA. So either from your blood or from your salivas, your DNA can be extracted, and then it can be put into the sequencer. Today it's parallel sequencer. So within one day, your DNA is fully sequenced. And then, thanks to big data analysis, AI and machine learning, we can simply run it through some model. And if this model is trained, or for example, traits that cause high blood pressure, or it can tell you if doing, let's say, weightlifting training, like strength training or endurance training, that you are, for example, better fit for endurance training. So it's definitely providing you quite interesting insight what to do to be better. Now, is it possible in Czech Republic to get your DNA sequenced? Yes, it is. And there are, at least to my knowledge, two possibilities. Right now, it's Slovakian startup called DNA Era. In case of DNA Era, they are sending your DNA samples under ID into German laboratory. And if you are rather thinking about doing something fully locally, then GHC Genetics is based in Czech Republic, and I believe it was originally established in USA, and they have their own laboratory in Czech Republic. Here we can see the picture how the transcription of the genes can then encode some amino acids which are then created. Okay, so let me go a little more up to explain you the letters that we've saw on the picture. So there are 4 basic nucleotides that are coding genes inside DNA, and they are called adenine, cytosine, guanine, thymine. And because DNA is helical, these bases combine together with each other. So adenine, which is marked as A, binds only with thymine as T, and cytosine with only guanine. So here these bonds would be in between the bases, and then the bases are transcribed as A, C, T, G. So when you then sequence DNA, you would be just writing these letters, and as it's mentioned, because the DNA is helical, on the other side of A, which is adenine, would be thymine, because adenine binds with thymine. And here you will get some line of letters, and then these letters are actually used to tell cell how to create, for example, amino acid. So let's conclude. Of course, you can go through the article to get all the details. So what is it all good for? It's definitely not going to relieve us from doing blood samples, but it can provide us with many interesting insights. So let's say that you feel tired for a long time, you have no idea by what it can be caused. You also visited your GP, also your GP did some tests, but there is nothing obvious. So you let your DNA to be sampled, and you figure out that, for example, you are in higher risk of evolving or having the lactose intolerance, which would then create inflammation in your body, which then deplete your energy reserves inside your body. So with this information, you then go to your GP and you do a precise blood samples for inflammation markers, and then also the lactose intolerance test. So instead of trying to search in a super huge area, thanks to the DNA test and sequencing of your DNA, you now have precise points to check, and you can check them, or you can also change how you live. For example, if you are at the risk of evolving high blood pressure, then you, for example, start using less salt, and that's it. So it can definitely help a lot and then prolong your life, so you can be around for your loved ones longer. And that's it. So definitely if you are interested, feel free to check the article.